All right, let's try it now. All right, that looks like that's working now. Okay. Yes, yes, yes. All right, awesome. Share my screen. Get that out of the way. Cahoots. Library. Yeah, we'll go over basically like the whole course, the whole setup of the course, as well as like kind of how to study for the exams and things like that. We'll go over that in the lab. I'll set, that's what I set that time aside for. So we'll do that for sure. So that will also probably help clarify some of that stuff for you guys that are a little unclear on that. Okay, I always forget the. There we go. Yes. Last lesson we talked about the same. I have a quick question. In Setman, we did your multiple jobs. We did do the blue change. Because it's moving based on what we learned last, probably positive because it's a clear background with just the. Right, you would see the cells being blue. Yeah, and so like that's how we see like toxicity, stuff like that. Yeah. Is that an example of that? That is a perfect example of a simple stain. Yeah, because you're just using one stain just so you can see the cells. Simple stain, probably methylene blue is what I'm gonna assume that it is. And we actually will be using that in our lab as a simple stain too. So yeah, it's a great example for that one. Okay, I was wanted to try to get some real applications. Yeah, I understand. And yeah, and some of the stuff too, like especially from chapter 3, it will make more sense once we get into the lab and we start doing it. And luckily, a lot of the basics for that stuff, we will have that under our belts in the lab before we get to unit 1 exam. So there's that too. So yeah, that should help some. Yeah, that's the only thing that has to yeah, the media selective and differential for now. All you need to know for the unit 1 exam
It's just the theory, the concept of what they are. We'll talk about examples and like literal like chemical makeup of some of those whenever we get in the lab in exercise 12, I believe it is. We'll literally be looking at some of the specific examples of that. Now, it just had to be spaced out the way that it's spaced out because of the timing and everything. But for the unit 1 lecture part of the exam, all you will need to know is the theory of that. There might be questions on the exam where I will say, you know, there was a lab worker at the hospital lab that got a swab sample from a patient who you suspect had MRSA, and they swabbed it on mannitol salt agar, which is one that we kind of reviewed a little bit over, right? I will not just say mannitol salt agar and then expect you to know what that is. I will, in the question, tell you it has salt to inhibit gram-positive bacteria, and it has a pH indicator that will result in different colors based on something that it produces as a metabolic byproduct. And so you should already know that the salt would then be selective, and then the color changing of the pH would be the differential aspect of it, which would make it both, exactly. So I would tell you that, so I will let you guys know that I might have examples of them in the test. There's no point really in studying like the exact literal examples of them because I will delineate them on the test. I'll explain them to the level that I expect you to understand them. So.
Her lecture quizzes kind of like a good like what kind of questions
there will be some questions on the exam that it will be very similar to the lecture quizzes. There'll be a handful, and I do mean just a handful, that might be the same question and the same choices. Like I said, a handful, and I will not tell you which ones those are going to be. Okay, you need to be prepared. So yeah, there it is to the degree of questions that are at that simplicity level. Our quizzes are easy compared to some of the questions that we'll have on the exam. When I say easy, to me, that's not a fair assessment. To me, the questions on the exam are just as easy, but there's more. My exam questions will have like kind of like what I just told you guys, that example of like, oh, the lab swabbed a media and they used mannitol salt agar, which had blah, 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 blah things. And then I'll ask a question relating to that info that I give you. You have to be able to pick out the useful info in that statement. To a lot of people, that's hard. It shouldn't be, but it is for a lot of people. Because usually, most of the time, you guys come from classes that are giving you this, what's the definition of this term? Or here's a definition, what's the term for it? And that's what you're used to getting on your test. Or here's an equation, balance it. Okay. So I want you to actually be able to pick and choose the useful information out of there. I'll tell you guys, and I cannot stress this enough. When you guys get into nursing, they're going to give you questions with answers where all of those answers are correct. All of them. You have to know which one is the most correct. I'm not doing that to you, but I'm trying to meet somewhere in the middle. So hopefully you can ease into that a little more comfortably. But if you guys can't do problem solving and picking out what is the important stuff and being able to recognize like this general thing, it adds up to this general thing. In nursing, that's going to tank you fast. That's actually probably the number one reason why most people don't make it through the first couple of semesters of nursing. They're just used to that black and white, right and wrong thing. You need to get over the right and wrong thing. Okay. A lot of the questions that we're going to be working on in the lab, on those, it's the ones that we're going to talk about in lab, but the lab worksheets that we work on in lab, yeah, there's going to be questions like, which of your organisms was positive? Easy enough, you'll know which ones are positive. That's going to be black and white. But there's also going to be questions like, tell me about what this tells you about your organism. That's a super vague question. And as long as you're in like the ballpark of telling me about what it tells you about your organism, there's not really a right answer or a wrong answer. I cannot tell you how many students have come to me and been like, how am I supposed to know what you're asking for specifically for the answer here? Okay. Grow up. The real world doesn't work that way, especially if you're going to be dealing with patients that have the way they describe their symptoms might not be the way that your textbook literally defined it as, right? And the way they're experiencing might not be the way that you would have described it. So we have to get past that black and white thinking. We have to start being able to pick and choose from our questions the important information and stuff like that. I'll try to give you guys some examples. We'll have one very specific example when we get to chapter 7, which is our last chapter before unit 1 exam. I have a very literal specific example. It won't be directly on the test, but it's a very good example of the type of questions that you may be asking test. And again, not all of them are going to be like that. Not all of them will be just what you're used to, terms and the definitions. and again, not word for word from what the study sheet, but very, very, you should be able, if you speak, and can understand words on a paper, then you should be able to figure out what the answer is. So there's that, the obvious kinds of questions. And then the ones that I'm getting at, which are critical thinking types of questions, where I will give you a whole slew of information and then ask you about one bit of that information in a way that you need to be able to interpret it and figure out what I'm talking about there. Okay? Like I said, chapter 7, I have a literal example for that. And I'll try to put together for you guys some other examples without like giving away the exact questions, because the whole point of this is to get you guys thinking in that regard and not giving it away, right? If I give you the exact questions and the exact answers, you might be sitting there and like, yeah, but that gives me the example of what it's going to be like. But if you just remember that question and that answer, it's back to the same start. I don't want that for you guys. I want you guys to be using your brains and thinking and analyzing and applying. So I'll try to work on giving you guys like maybe some examples. I don't put them on the quiz on purpose because I want you guys to be able to see the quiz as like that intermediate. I don't want the quizzes to be too hard, but I do want you to be applying and looking for and realizing how the information fits in with one another. When the quizzes kind of be easier points for you guys. But still, you have to know. If you've done the quizzes and you know what I mean, like you have to be ready for the quizzes because it is timed. So you have to have your notes ready and you have to know where to look in the notes and how to look in the notes and things. But I also don't want to put too much pressure for you on the quizzes with like really hard critical thinking questions that might take more time and all of that. So I'll try to see if I can put together like 5 or 6 questions that are similar to the critical thinking, so you guys can get your feet wet and see what it's going to be like. Because I'll have questions like that on all of my exams, the critical thinking ones, and it's not the majority of the test, but you will see probably between 5 and 10 of those questions will be those types of questions. I want you to prepare for them. I think they're actually the more important questions than the obvious ones. So be prepared for that. I'll try to put some things together for you guys. I handwrite all of my questions. I don't pull them from a bank from a Rocket or something like that. So I try to be thoughtful with them. So it takes me time to put those together and have like new ones that I have separate for you guys. I'll have to actually sit down and write them myself. So we'll have that. But I try to put them in a perspective of like real world application too. Which I feel like lacks a little bit in like the 1000 level classes like chemistry, where they're just trying to get your basics cranked out. And you get into classes like my class or physiology, where we're trying to put a real world spin on that, and you guys aren't used to having it put in that perspective. But everything you're going to do in nursing school, I know not all of you are going to nursing school. I know some of you guys are going to vet, but or maybe even pre-med or something like that. But the idea is the same. I want you guys to be able to relate it to real world. That's more important to me even than you guys memorizing the terms. Okay, so for example, one of my best students that I've had works her butt off to be the best student. She's not one of people that comes easy necessarily. Worked her butt off to get her A. She came to me, it's her first semester, just like you guys, first week of nursing school for her, right? She comes and she's like, already in the first day of her classes, they've talked about 6 different topics from all over microbiology. And I have so many students that come to me to treat this class like just like another stepping stone. You're not going to actually use it or whatever. First day. Don't kid yourself. Don't lie to yourself. Don't be in denial. I have students that are in nursing school. I literally think they're just in denial about how much they've carried from this class, and that they actually, when your advisors start talking about C. diff, when your teachers start asking you about MRSA and things like that, you'll have more knowledge that's just there in your brain from having been through this class. You don't realize that it already helped you understand that thing. You might not be actually sitting there talking about the molecular makeup of the cell wall of the bacteria, but that basic knowledge will help you in understanding how to better treat your patients and why that doctor is prescribing this antibiotic as opposed to other antibiotics and things like that. So, and what an allergy is, what's really going on in your body when you're allergic to something, like that. We'll talk about that in this course, and all that. So I like to put that real world spin on things. I think that's actually really important, and it lacks way too much in courses that are leading to this one. But yeah. So I hope you guys kind of put a little perspective on that. And like I said, I'll try to get you guys some examples, like just for your own practice purposes, so you know what I mean. But it really is a lot like that, where I'll be like, give you a scenario of a patient, and then all this information that you get from that patient, and then ask you a question about one aspect of that, and you'll have to be able to pick out what you need from that to answer the question, or something that the lab is dealing with that I would still expect you to understand for our course, right? So that it makes sense to you when you go into the field. I'll write some questions for you guys so you have an idea. But yeah. Any other questions before we get started with the Kahoot? Everybody ready for the Kahoot? Signed in? Keep going. Like I said, for the first probably month, I'll walk you through these questions when we do our reviews. And then depending on how it looks, we will probably play games instead. But I want to be sure we're getting a good idea of what these reviews are for. So we're doing chapter 3 because I felt like it had the most content compared to chapter 1. It was mostly just. All right, which of the following is not included in the 5 I's? All right, so the answer for this one is infection. So our steps would be inoculation, incubation, isolation, inspection, and identification. One of the best things you guys can do when you're studying for your exams when you get to that point is I'm going to open up your quizzes here. I have to go through it. It's one of the things on my list that I haven't gotten to. But to have it where like when you guys take your quizzes and you're done, it doesn't just give you a score, but you can go and see which ones you got wrong. I'm not ever going to open a quiz or a test and tell you what the right answer was. You need to figure that out for yourself. What's the right answer? Why did you get this wrong? Where was the right answer in this? Because I feel like one of the most valuable things you can do when you're studying is understand why the wrong answers were wrong. Even if you got it right, just because you like memorize the 5 I's, let's say. You're like, I memorized the 5 I's. I know this wasn't one of the ones I memorized. That's fine and that's a good start. But if you don't understand why infection wasn't one of the 5 I's, you got a problem. So when we're doing the 5 I's, you should also be thinking about it as the 5 I's are the steps that are involved for figuring out what the organism is that's involved in a disease or just in general what an organism is. And so is infection an important step in figuring that out? It's not something that you're going to do to figure it out. It might be something that your patient has when they come to you, but it's not one of the steps you're doing to investigate it, right? That's why infection isn't the right answer. If you can't think to that on your own, that's what I mean by understanding the material. Make sense? Okay. Makes sense now, right? But then when we get down to doing it ourselves, keep that in mind. All right, differential media, what is it? All righty, so differential media highlights metabolic and biochemical differences. Those should be the same thing, honestly. The biochemical processes that go on in your body, that's your metabolism. All right, so differences, those differences between organisms. In the media, when you're looking at that, that's usually defined by color change. But what does that color change represent? Metabolic and biochemical differences. That's what the color changes represent. It's actually more important that if we see a color change in our media, that you not say, oh, it's pink, that's positive, that you understand that if it's pink, that means it was an alkaline pH shift because it broke down this, that, and the other, made these products, and so on and so forth. That's what it should be in your brain when you look at these different differential medias. Like I said, with differential and selective media, I don't need you to know the exact examples in like memorize them in your brain because I'll give you that info on the test. But if I tell you mannitol salts agar was used by the lab, it had salt to inhibit gram-negative growth, and it had a pH indicator if it was able to ferment the sugar. What type of media is it? You would say it's both selective and differential. Or if I were to ask on the question instead, which aspect of the media made it differential? You should be able to tell me it was that color change with that pH difference, dependent on that metabolic. I could ask about any of that, right? That's how you should be looking at that stuff. Starting to see how it might be a little more complicated than just remembering the terms. All right, selective media, the other side of it, right? Make sure you're reading your choices. A lot of my test questions, when I write this seems super obvious to me. Be sure you're reading your choices and you're understanding each of your choices before you make your selections as well. All right, so our answer here, selective media. Selective media is the one that will select for some organisms. We can term it that way, but another way of wording that, inhibiting growth of some organisms, encouraging growth of others then. Same way of saying selecting for some. This is where I will tell you, this is where that word for word understanding of the definition is not going to help you. Because I can stand up here and say it selects for some organism, that helps you remember it. If I were to say, you write down on your scratch sheet when you get to a question about selective media, select for some organisms. If you don't know what selects for some organisms means, it's not going to help you if I don't use selects in your choice. You better understand what the words you're writing down mean and what I'm telling you, what it actually means. So that's why, most of you guys got that one. That's why that's the right answer. We're getting that. We're starting to see. We're getting a taste of it. What type of media supports fastidious bacteria? So we had three terms that kind of related to one another. One term described the media. One term described the bacteria, which was obviously fastidious. And there was another term that described the thing you put in the media that helps the fastidious bacteria grow. So the term for the media, when you add the growth factor to it, that the bacteria needs to grow, your fastidious bacteria, the media that you added the growth factor to is called enriched. That's a term that we should be familiar with. I know it's just like review and you may not have reviewed for it, and that's fine. This is just participation. So if you're not getting them right, that's okay. That's why we review. All right, streak plate technique. What do we use it for? All right, the whole point of a streak plate technique is to spread out those cells individually so we can get them spread apart as individual cells, which will then grow up into individual colonies. Each colony came from one cell, and so you can take that one colony with your little sterile loop and put it into a sterile media that it will grow in and represent a pure culture. The whole point of that is to isolate those organisms. Alrighty, the diagram will show a light microscope. Name the part of the microscope labeled X. All right, so that is the objective lens. This one, you gotta know the microscope parts. True or false? Stains improve contrast. Is that what stains will do for us? Yeah, that one's true. Pretty straightforward one. The whole point is if you were to look at any of these cells, whatever kind they are, it doesn't matter. Cells in general, if you're not staining them, it's like looking at a colorless thing with a colorless background. It's like almost impossible to see cells if you don't add something to it for contrast, and stains are what we use for that. You'll see what I mean then. All right, the Graham stain is a type of differential stain. This is actually a picture of a mixture of different kinds of bacteria with a Graham stain. We had a list of the different kinds of differential stains. We didn't go over them individually, and that's okay if you don't know them right off the top of your head now, because we're going to go over them in chapter 4 in detail. So you will get them for sure. Which part of the microscope generates the real image? So we had one area of the microscope that you went through that created a real image, and the other part of the microscope that I went through that created the virtual image. Which one creates the real image? All right, so this is going to be the actual objective lens. I know, so if you guys are taking the optical lens, it's easy to think that we're looking through the optical lens, and so what you're seeing must be the real image because you're seeing it and you're real. But the way that the theory of it works is that the light shines up through the bottom of your slide, through the specimen, and hits the first lens. The first lens that it hits creates the real image. That's the objective lens. It makes sense when you follow the light flow through the microscope. And then it goes through a whole other lens. So we're distorting that image yet again. That's a virtual image that hits your eye. We've actually magnified it to the point that it is like 1,000 times bigger than real life, quite literally. So it's not the real image that we're creating there, but the virtual one. So virtual, like I said last time, virtual, think virtual visual. That's going to be the ocular eye visual. All right, what is the function of immersion oil? All right, so immersion oil, that's the one that we put on the slide, between the slide and the objective lens, that prevents the light from just scattering around from hitting the air. We go through the glass. If we hit the air, the light will diffuse just like it does from light bulbs in the room. We want to keep nice and tight. We want to keep all those light rays. Each of those light rays, you can imagine, represents some characteristic of that image. The more we keep into the actual lens, the more detail you're going to see. Refraction is that dispersion of the light rays. That's the term for that. So we're reducing refraction. Another way you could word this, think about that in your head. How else could you reword that correct choice? Reduces light scatter. It's another way you could word that. All right, here's our podium. So this is one of those perfect examples that I could say where when people ask me about like the quiz questions or the Kahoot questions being a good example for what's going to be on the test. To me, they are. They're not going to be the literal approximations, right? But if you can look at these and understand why you're choosing the choice you're choosing, not just from memorization, but from understanding, then you're preparing yourself pretty well for the test. But you have to look at it from that way. Right? I hope that helps you guys kind of get the perspective a little bit on how to approach these things. All right, so let's move on. Good job, guys. Honestly, first one that actually was like about something. You guys did pretty good. Really, login expired. I don't even know how to get out of that. I did enter the duo code, I promise. This computer's great. I have to do this every time that I use this computer. I have to like verify that I can use PowerPoint. Wonderful. 854. It's the only one that's on campus that's like this. All right. So we're going to get into chapter 2 today. Chapter 2 is basically a chemistry review. I'm going to go over the aspects of chemistry that are kind of important for this course. And yes, like I said, like I kind of warned you guys, we're definitely going to be using chemistry in this course. Almost every day, I will have some aspect of chemistry that will be incorporated in with what we're talking about. And I'm just probably going to be throwing these terms around left and right like salt and pepper, man. So be sure that these terms are familiar to you. If I start saying like the reason that prions aggregate and cause problems in your brain, poke holes in your brain, is because they are exposing their hydrophobic regions. And those hydrophobic regions don't mix well with the water in your body. If hydrophobic is something that just flew over your head and you're not really sure what that really means in the terms of these molecules, time to start clinging on to that stuff because we're going to be using terms like that all the time in this course, okay? But that's what this chapter is for, to give you the review of that stuff. Talk about it and put it into our own perspective. All right. Also, before we go on, here's the people that I rolled for today. In case you can't read this, I'm going to tell you in order when we get to those last 5 questions. We did get to them in my last class, so we'll probably get to them in this class. Okay. I'm going to try your name. Sheet Lalik. Is that right? Seek Lalik. Okay. I'm probably going to get it wrong a few times before I get it right. I apologize. All right. Then we have Brittany. Then I'll do number 3. Then we have Alanis, and we have Kayla. Everybody here? Yes.
What does that mean?
Okay, so this is the last 5 slides of this lecture. We got to them last time, but I had to cover them because we didn't have time for everybody. For those last 5 slides of my lecture, and they're on your slides on campus. So they should be available to you. So if your name is up here, you can prep for your question. it'll be in order. So 1, 2, 3, 4, 5. I will ask you your question and you'll answer it in front of the whole class. Okay. And you prep for it. If you don't want to answer your question, today's not your day, or you didn't want to prep for it or whatever, when I come to you, you can pass. I will answer it. If you pass, you get 2 passes a semester, and I will keep track of those. So keep that in mind. You get 2, but then if you come a third time and you want to pass again, then what this is, as long as you don't exceed your 2 passes for the whole semester, you get 1% extra credit tacked onto your course. You exceed your 2 passes, you forfeit your extra credit. Okay. So this is just encouraging you guys to participate. I said in the last class, and I'll remind you guys again when we get to those questions, as long as you're in the ballpark of the discussion, I'm going to count it. As long as you are participating in what we've been talking about today, you're good. Just show me that you've been paying attention. All right. And if you don't know it, or you have a question about it that is intelligent and thoughtful, I'm good. All right. We should by now probably be aware of atoms and molecules, but just to remind you guys, atoms are what make up all matter in the universe. And those atoms consist of a nucleus that consists of protons and neutrons. And around that nucleus, the protons being positively charged, the neutrons obviously neutral, and electrons having a negative charge. And this negatively charged electrons being held in towards that nucleus because of the positively charged, balanced out positive charge of the protons in the middle. Those negatively charged electrons floating out. It's more like a cloud than it really is those orbitals like they try to show us that they are. But in each level, each shell that we have can house like a favored number of electrons that I'm not going to quiz you on or make you know. But as long as you're aware of the properties of an atom and the general, function of how that exists, that's going to help you go a long way in understanding a lot of these molecules. So the electrons are out on the outside, they're hanging out in the correct orbitals, and some of those orbitals would rather have a certain number of electrons in them. And once you get past that second one, it's going to be like 2, 8, 8, and so on and so forth. All right, so terms to be aware of in chemistry: a molecule is any time that you have 2 atoms together. So a compound is when you have a molecule that's 2 different kinds of atoms at minimum. So if you have H2O, that's a compound. It's got hydrogens and oxygens. Whereas oxygen, O2, it's a molecule, but it doesn't qualify as a compound. All right, there's 3 different kinds of chemical bonds that you'll have to know and be aware of, and I will test you on the exam. This is going to be covalent, the ionic, and the hydrogen. With covalent bonds, we are sharing our electrons. So on those outer orbitals, maybe we each would like 1 electron, and we both need an electron, so we can just share an electron between each other to keep each other happy. Let's imagine that you have a younger sister. So I'll do myself. I'll put myself out there. I have a younger sister. My sister's name is Erica. And so my mom might buy us 1 Barbie. We might have 1 Barbie, and between us, we have to share that Barbie. So Barbie might be the electron in this case. So we have to share that Barbie between us. If we're nice, good kids, then we're going to share it equally, and nobody's going to be hogging it. Unfortunately, in chemistry, that just isn't the way that it works. And of course, I'm the older sister. Do you think it works equally between the 2 of us? No, of course not. Of course, I was hogging Barbie. I'm not going to lie. So covalent bonds are important in the fact that we are sharing that electron, but the fact that somebody might hog it more than the other guy gives a unique characteristic to some atoms. We'll talk about that in a second. But covalent bonds, we're sharing those electrons. In an ionic bond, let's say we have Barbies, and I have one. I have enough. I don't need this one. So I just give it to my sister so she can have it, and she can keep it, and it's hers now. It was mine originally, and now my sister can have it. Or think of hand-me-down clothes. This shirt doesn't fit me anymore. Now you can have it. We're not sharing the shirt. It's hers now. I gave it to her. So ionic bonds are when you donate or transfer or give an electron to another atom. We see this a lot with things like sodium and chloride. That's the best example I could probably give you in pictures. That'll probably clarify that a little bit more than me just explaining it straight up. But I have an electron that is like in an extra orbital. Yeah, I have the number of electrons that match my proton charge, but that extra orbital guy, he'd be happier just not being there. And there might be another atom out there that has 7, would rather have 8. Yeah, their electrons matches their protons in the middle, but that'll fill out that 8 and make that orbital happier. So then we can I'll give up my outside electron so you can fill out your orbital. Now I'm positive, and then they have a negative charge because the electron transfer. That's an ionic bond. I'll show you a picture of sodium and chloride in a moment that will help clarify that. Then we have hydrogen bonds, y'all. It's a little more complicated. So hydrogen bonds, genuinely speaking, like this is one of the ones that trips a lot of people up, because it's very important that we understand how our covalent bonds work in order to get how, I used to have a tablet I could write on up here, and now I don't know where it went, but I'll do the best I can with my mouse. Okay, so please forgive me. But basically what you need to understand is that hogging aspect of covalent bonds. Anytime that you see a chemical structure that is drawn with lines like this as demonstrating the bonds, those are covalent bonds, always. Okay? Might be simple enough if you come from chemistry recently and that makes sense. But those are covalent bonds. That means that we are sharing electrons, always. So we're sharing electrons between hydrogen and oxygen in a water molecule. Two H's, one O, H2O, this is water. Oxygen is something that we would call electronegative. I'm not going to write it out on here because that would be the sloppiest handwriting you've ever seen in your life. But the term for it is electronegative. And I think I have it on a slide. Let's see. Talk about the covalent bonds. We talked about sharing. Polar. Yeah, here we go. Okay, this is good. So oxygen is very electronegative. It has the, like I says here, a nice way of putting it, the ability to attract electrons. It hogs electrons. It's me. So if I was oxygen and I had two sisters and I was just hogging all the Barbies, that's oxygen. I want the Barbies for myself. Okay, fine, I'll share with you if I have to when mom's looking. But otherwise, they're my Barbies. I'm hogging them. So that makes oxygen have a negative polarity. So you could see it as being like sort of like a negative charge. It's not separated. It didn't steal it completely. We're still having to share those electrons sometimes. But overall, generally speaking, the electrons are spending more time with me. The Barbies are spending more time with me than they are with my sisters. Okay. So oxygen's hogging because it's electronegative. That gives a negative charge. But because I'm hogging them so much, and they're not spending as much time as they could with the sisters, the H's. What charge does that give the H's? Positive, right? Because they're not seeing the Barbies as much. They're not having the electrons around as much as they should. So they get that positive polarity as a result of that, okay? So that's important to understand because what's the solvent of life? Water. There's a reason for that. It's that polarity, okay? So we're going to talk about that quite a lot. That's really important to get. And you probably talked a lot in chemistry also. Sort of like if you guys heard that, you heard of Schrodinger's cat. Probably it's a good thought experiment of like, if you have a cat in a box, is it alive or is it dead? Well, how would you know? You would open it up and look. Right, because once you open the box, it is alive or it's dead. And it's one or the other. But until you open it, you can't know if it's alive or dead. So it could be both at the same time simultaneously. It's kind of like how water is, okay? So water might exist as we think of it as H2O in liquid. I know you guys probably talk about this in chemistry. I don't sit in on Dr. Kim or Dr. Singh's classes or whoever adjunct you might have had, but I'm sure they brought this up, right? You've probably seen this nonsense. Oh, be patient with me while I click this out. So we'll have H pluses and we'll have OH negatives. So kind of simultaneously the whole time, you'll always have H2O. Sometimes that oxygen gets so greedy that it just steals that electron from one of those hydrogens. And it kind of exists in that state simultaneously at all times. You can always think of it as being that way because that oxygen will get so greedy. So you guys remember what these are, right? We have our protons or hydrogen ions. You can think of them that way. Or you have your hydroxyl groups, right? So if you have water and it's just water, it's pure water, all that's in it is H2O. There's no other thing in it, okay? So if you have water, you would have to imagine, because of math, that your H's and your OH's, if we were to dissociate all of those H2O molecules, the H's and the OH's would have to be equal because math, right? So if your H's and your OH's are equal and you have equal amount H's and equal amount OH's, well, if this doesn't lead you up to the reason for 7 being the pH of water, I don't know what it is. Because what does pH range from? 0, does anybody remember the top? 14. So why is 7 in the middle? Because the H's and the OH's equal each other. It's right smack in the middle. That's why water's in the middle, because you have equal H's and OH's. That's why. Okay. Should be equal. That's why that's neutral. Neutral, N for neutral. All right. Really important that when we think of pH, we're not thinking of it being near the 0 or near the 14, that we're thinking of it in terms of relation to the 7, right? Your body's pH is not 7. Your body's pH, does anybody know the pH of the body? Right, around 7.35, 7.45. And that range is actually incredibly important. If you go to the doctor and your pH is 7.48, they'll actually be concerned. Even that little shift, they'll be concerned. So pH is so important in your body because of maintaining the balances of those H pluses. But why is it important that we maintain the balances of those H pluses? That is going to lead us into that conversation of what our macromolecules are, what proteins are, why they are the way they are, and how they function, and all of those things, which we'll get to at the end of the chapter. But I want to build into that so you understand the importance of understanding this. I'm not just talking about pH because I have to. I'm talking about pH because this is going to apply to everything that we're talking about for the rest of this course. This course is big picture ideas. We've ranked up our little picture stuff. Now we're going to apply it to the big picture stuff. All right, get out of here. All right, so we have that picture that I was showing you guys about ionic bonds. This is the picture of the sodium and the chloride. See how sodium has this extra guy just out on its own little orbital here? It's not so keen on that. That electron that's out there for sodium, it's not so keen on staying out there on its own little orbital. Chlorine, it has 7, where it has an open space here. It'd rather have an 8th. So it steals from sodium. Sodium's happy to give it. So what does that make sodium? Positive charge, because now its electrons and protons don't equal out. There's more protons than electrons when it does that. It donated, it transferred, it gave its electron to chlorine. Think of chlorine as my little sister. When I grew out of that shirt, she can keep it. Okay. So that's what's happening here in ionic bonds. And so these guys will exist as in compound form as just sodium chloride. And they've already donated those things. But now when I'm introducing these guys that have already donated their electrons to one another, because that's how they are as a compound, when I put them in water, which is polar, what do you think the sodium is going to do? And what do you think the chlorine is going to do? They're going to, the positive associate with the negative oxygen, and the negative chlorine associate with the positive hydrogens of water. And that's part of what's going to get into that conversation of how water is so important as, our solvent of life. So that process of those guys kind of being able to donate those electrons and then associate with other polar molecules in whatever mixture they're in, that's ionization, when they separate out and can interact with other things, especially water. So here's a picture of that. Essentially those chlorines, which have their negative charge because they stole that extra electron, being happy to associate with the H plus polarized hydrogens of water. Make sense? Okay, so that's important. We know sodium chloride is important in our bodies as well, because it's going to help maintain our salt content in our cells. And if you guys remember about learning in chemistry about tonicity, like hypertonic and hypotonic and all of those terms, we aren't going to talk about those in this chapter. We're going to get to those in chapter 9, but that's going to start that conversation as well, right? So having that balance of our salts versus pure water, that stuff's important too, not just pH. All right, so hydrogen bonds. So let's talk about it. Let's see if I have a good picture. I do. Okay, let's just move on to the picture. So in hydrogen bonds, we introduced this concept of water looking like this, right? We have our hydrogens, which have our polar positives. Because the oxygen is hogging away those electrons, it gets this little polar negative. It's electronegative, hogging the Barbies and whatever. Because it is electronegative, it is happy to associate with positively charged molecules. So that could be the positive hydrogens on other water molecules. It could be the positive sodiums if we're adding in sodium chloride. It could be H pluses from adding in acids that donate H pluses, or from the H pluses that might be dissociating from water when we have the H plus and the OH negative. It doesn't matter. Oxygen being negative is happy to associate with positives, and that's great. But a hydrogen bond specifically is whenever you have a positively charged polar positive H plus that is attracted to anything negative. Now, in this picture, we have this H plus here, and this negatively polar oxygen here, and these guys are attracted to each other. So this here in the middle, that's the hydrogen bond, okay? Is there any actual electron exchange going on between that hydrogen and that oxygen that are hydrogen bonded together? It's just attraction. There's no electron exchange. It's important to remember that. It is just attraction, okay? So our covalent bonds, do we have movement of electrons? Yes, we're sharing the electrons. In ionic bonds, did we have movement of electrons? Yeah, we donated or transferred those electrons. So our third bond type, hydrogen bonds, is there movement of electrons? Nope. We have to have had a covalent bond where we had hogging, yes. But the actual hydrogen bond itself is there? No. Okay, just attraction. Really important that you remember that. Because if I were to draw you guys this. Wow, this is really, really artistic rendering of it, but this as an unwinded version, this is gonna make more sense when you see this one, of this. What's this look like to you guys? Right, pretty crappy rendition of it, but you get the idea. So two sides of a ladder. And on this ladder, we have, basically it's gonna be a sugar molecule. We're gonna talk about the parts in a moment, so don't focus too much on it at this particular time. We have a sugar molecule, which I'll put a circle there for it. We have a phosphate molecule, which I'll put a P for, and then we have a base. And this whole structure just right here is a nucleotide. We'll put NT for nucleotide. And on the other side, it's the same thing. We'll have a base, we'll have a sugar, and we'll have a phosphate group. They go in opposite directions. We'll talk about why that is in another chapter. So it's like this. How are those two bases linked together in your two-sided ladder for your DNA? Do you guys know how? Hydrogen bonds, right? Your entire double helix, each side of your double helix is held entirely together just by hydrogen bonds, just attraction. There's no electron exchange, it's just attraction. So if I'm telling you that it's the ability of, it's going to be oxygens, it's 99.9% of the time oxygens, it's the negative, okay? And then the positive has to be hydrogen. That's what makes it a hydrogen bond, right? So we have hydrogens that are positive on one side, they attract to oxygens that are negative on the other side, the other base, A to T and C to G. But that's what holds together your entire genome. That's it. And so those attractions are what's holding that together. In fact, that's often what holds together a majority of the shape of your proteins that are doing all of that work for you in your cells too. So that's incredibly important that you get that, because once I start throwing off your pH, I start throwing in extra H pluses. Guess what? Those guys are going to love to interact with that oxygen instead of the H across from it. Now you're dissociating your entire genome. You throw in too many H pluses. You throw in too many OH negatives, they start interacting with the H's. We start shifting that balance of the pH. Suddenly your DNA can't hold itself together. Suddenly your proteins can't hold their shape and do their job. We're starting to get the importance of pH now. It's not just a theory that you're talking about in chemistry, it's what's holding your body together right now, literally. So you get it. All right, that's the importance of a hydrogen bond. Probably the most important concept that you can get as your framework, forgetting why all the other crap I'm going to talk to you about is important, is a hydrogen bond. Understand the concept of it. Right, so we know what a solution is. Just to review to you guys, a solution is when you have a liquid and something is dissolved in it. The liquid is called the solvent, right? And the thing that's dissolved in the liquid is called a solute. That's pretty much the only time you're going to hear me really talk about this. So don't talk about solvents and solutes a whole lot in this class. And I know a lot of you guys probably get confused between which one is the solute and the solvent. And I do too sometimes, okay? but here we brought it up because I have to. A concentration, that part's a little easier to understand. The thing that's dissolved in the liquid, how much is there in it? That's the concentration. That's a little easier. All right. Water, we know it's the solvent of life, and the polarity of it is kind of what helps us exist, and how our molecules are going to interact with it. And how molecules of our body, whether or not they have charges, positive or negative, if it has a charge, hey, there's probably some part of water that's happy to interact with it. If it has a negative charge, the hydrogens are happy to interact with it. If it has a positive charge, the oxygens are happy to interact with it. And we can fit you in somewhere. You don't have any charge at all, get out of here. You don't belong. So, being mostly water, molecules that can't interact with water are our non-polar molecules, hydrophobic, water-fearing molecules. So if I ask you, what type of charge does a molecule usually have if it is hydrophobic? You should be able to tell me it doesn't usually. It's non-polar, right? Because if it has a charge, charges go well with charges. If it doesn't have a charge, we're talking about oil with water. We're talking about fat with water. We're talking about wax with water. Those guys, right? You guys are aware of these properties that these things have. Be aware of what hydrophilic, water loving, what that really means. It means you have charges. Be aware that hydrophobic means water fearing. That's because you don't have charges. Amphipathic, anytime you hear amphi in this course or any other science course, amphi is going to mean both of whatever you're talking about. So in this case, amphipathic, we have part of it that is hydrophilic and another part of it that is hydrophobic. Pretty big one when we're talking about things like phospholipids, which I know you guys have been introduced to in the past. Super, super, super important molecule as far as cellular membranes go. We'll talk about them too. But so you need to know all these three terms. Hydrophobic and hydrophilic are terms I'm going to throw around a lot. Okay. We talked already a little bit about the pH scale. So we're going to come back to that now. I want to show you the actual, it's easier for me to explain this if I have the actual scale up here. So yeah, here's some words that explain it that are already typed out, but I'm going to use my own words to describe it to you once I finally get the freaking scale here. Okay, here we go. All right. So we can see here, we already said 0 to 14 is our scale. You just need to remember that one, okay? 0 is easy enough to know that that's the start, but just remember that 14 is the end of it. And if you ever can't remember that, if you can at least remember that 7 is the middle, you should be able to know that 7 is the middle. So 14 should be the total. Okay. Makes sense? Fair? So 0 to 14. These numbers, and what do they mean? pH, when it does that, just pretend like that didn't happen. pH equals the negative log, and I showed it on the previous slide in much better writing than my handwriting here, but I want on this slide too. The negative log of the concentration, that's what these brackets mean, of hydrogen ions. That's H pluses. So if I told you already that H2O existed as both just H2O with its polarities, as well as hydrogen ions, plus OH or hydroxyl groups, and pH is the negative log, we'll get to what that means in a moment, of the concentration of hydrogen ions, and our scale is 0 to 14, and you know they have to be equal if they're just H2O pure, then that is, again, coming back to that, I know I already said it, but to reiterate, that's why we're at 7 for neutral. Oh, it already says neutral below, but that's fine. There you go again, neutral. All right, so we've got that down. So what's really going on with these numbers? So pH, always remember that that H is literally referring to the H pluses. That's why the H is there, okay? So pH is referring to those H pluses. Log, this means it's base 10. Thank God for that. Honestly. Okay, so there's the base 10, that's what that means. Okay. The negative means we're talking about it being inversed. I'm not going to write inverse, I'm going to put INV, you guys know that means inversed, okay? So what that means is, if I were talking about 10 to the second power just by itself, that would be a 1 with 2 zeros by it. That's what the 2 was telling you, a 1 with 2 zeros by it. Okay, so it's 10 to the second power. Another way you could look at that is just 10 times 10. And wow, 10 times 10. It's telling you there's two 10s there, right? That's what that means. Just, I know it's basic math, but just helps you visualize a little bit what's going on. So that if I had 10, that was to the 14. I'm not going to write it out, but you guys get it, right? It'll be 1 with 14, zeros after it because you're having 10 times 10 times 10, or just 14 tens, right? Okay, so that's our normal basic understanding of what a logarithmic scale is. But this guy is inverted. So what's really going on in the pH scale, we have the basics of how that works, but 10 to the -2 means you're going to take a 1 and you're going to move it back 1. If you have 1, you would put it here. You're going to move it twice. So your decimal point's going to be here. So it's 0.01. That's really all that that's telling you. Or you could look at this as the fraction of 1/100. So all of these, 10, I didn't do that, just to be clear. 10 to the -2 is either telling you how many decimal points to move it, or it's just telling you that 100 that we got over here, put it under the 1, whatever your number is. Okay, so we've got that written out for us, so that if you did have 10 to the -5th, you would have 0.1, 2, 3, 4, 1, or 1/1, 2, 3. That's right, right? 1, 2, 3, 4. You guys see how good I am at math? Okay, so you guys get this, right? Like where this is coming from? Okay, so you guys can now see that, that 10 to the -2 is a bigger number, easy to look at it now, than 10 to the -5, which is when we talk about pH, you're actually looking at it that way. So pH of 2 means 10 to the -2. pH of 5 means 10 to the -5. Okay, so pH of 2 means you have more hydrogen ions, because that's literally the concentration of the hydrogen ions. Your concentration is this, or your concentration is this. Literally. Okay, so that's where those numbers are coming from. Does that help put back into terms? I know you've learned this before, but does it help remind you guys of what the pH scale is and how it works? Does it help a little bit? We all clear on it? Does anybody need to clarify it any more than that? It's like, I don't want to say speak now, forever hold your peace. You guys can always email me if you get confused about it, okay? All right. Because on the test, I will not just ask you, is 2 more acidic than 5? But I might ask you, which number has the higher hydrogen concentration? I'll say pH of 5 or pH of 8.73. Which one has the higher hydrogen ion concentration? 5. So if you need to write out the scale on your scratch paper or something like that, do it. Whatever helps you understand what's going on with the actual hydrogen ions in a pH scale, okay? Because like I said, this is telling you how many hydrogen ions are going on with your situation. And how many hydrogen ions are going on with your situation is important for the structure of all the molecules in your body, because those hydrogen bonds are what's holding everything together and helping everything function. And so you can start to see how you can change the shape and function of molecules based on shifts in pH even. Cool. Big picture stuff, y'all. All right, so reminding you guys of what organic molecules are. For the purposes of my course, what I need you to know is, of course, organic molecules have carbon in them. That's the basic of it. But if it only has carbon in it and there's no hydrogen in there, it's not really truly organic. So carbon dioxide, not organic. Calcium carbonate also has carbon in it, not organic. They don't have a hydrogen backbone to support it. So for organic molecules, we're looking for carbons that also have hydrogens with them. Carbon is the important one. If it doesn't have hydrogen in there, it's not truly organic. Cool. All right. Carbon's very happy. It's a very, very, very calm, nice, neutral atom. You give it, it has 4 electrons in its orbital. And that's actually not so bad for it. It's actually kind of happy with that. But you give it another 4 to fill out those 8, and man, is it happy. And it will share equally. It is that big sister that is happy to share with all the little sisters equally. Yeah, you can play with my Barbie. I had it for 2 hours today. You get it for 2 hours today too. or just play with the sisters, whatever it is, how you want to look at it, okay? So carbon is very, very good at sharing. Water, we have oxygen that's terrible at sharing with its hydrogens. So the hydrogens that are interacting with carbons are very different hydrogens than the hydrogens interacting with oxygens. The hydrogens that are interacting with carbons came from a very healthy relationship. The hydrogens that are interacting with those oxygens are used to having those electrons stolen from them all the time. They behave very differently. So our hydrogens that are associated with our carbons, they don't have a positive charge to them. They're very neutral. That's bad when we're mixing you with water. Water has all that polarity. When it sees things that don't have polarity, like we said, that makes them hydrophobic. The reason we call them hydrophobic. Oil doesn't mix well with water. It has a lot of hydrocarbon action going on. Everybody nice and neutral. We don't need that stability in our lives. We like the chaos. That's what water says, okay? So that's how that it would look at it. So cool. So we're starting to get that idea. And I'm not going to ask you too much about double bonds and things like that in this class. That was what chemistry was for, as far as I'm concerned. If you guys go take biochem later on in your life, come talk to me. I'll be happy to talk with you about it. But yeah. So let's get into the idea now, building up the purposes of these molecules in our big biological molecules. The purposes of carbons and oxygens primarily. Carbons that are going to be creating those nice, stable, neutral interactions versus the oxygens that are hogging as many electrons as they can hog. Those are probably the two most important aspects, and then the effects that they have on their hydrogens and how those hydrogens interact with other people. Right. So our major groups of biochemicals are going to be carbohydrates, lipids, proteins, and nucleic acids. So let's go through them. These are our macromolecules. That means they're big molecules made up of smaller parts. Those smaller parts are called monomers that come together to make polymers. Okay. For the carbohydrates, our monomers are called saccharides. Another word for a saccharide is sugar. Okay. So we have multiple sugar units linked up to one another to make our polysaccharides. Things like starch, where you have multiple sugars just linked together. That's how, things like potatoes are going to store their sugar for later use. I'm linking them together like that. So those are our carbohydrates. They're made up of sugars. Sugars, typically when we name them, they end in ose. So anytime that you guys hear me talking about a molecule that is blobody-ose, ose should tell you immediately in your brain that that is a sugar. Okay. We have hexoses that are 6 carbon sugars and pentoses that are 5 carbon sugars. Those are the two groups that we're really going to see the most of. Probably the most common 5 carbon sugar that we're going to talk about in this class is ribose, and its friendly derivative, deoxyribose. How many carbons? So I'm telling you that ribose has 5 carbons, okay, just to be clear, and you're going to need to know that. But if ribose has 5 carbons, how many carbons do you think deoxyribose has? 5. Deoxyribose. It's still ribose. What did we remove from it? Oxygen. Is that a carbon? No. So how many carbons does it have still? 5. It tells you in the name. Okay. You can remember ribose has 5. You've got deoxyribose down. Okay. So 5 carbon sugars. Right. Our 6 carbon sugar that we'll talk about the most is glucose. So those are probably the biggies that you'll need to know. I'm never going to have you draw them. I'm never going to have you recognize them and pick them out of a lineup as far as the pictures go. Okay. But you should probably get it under your belt that glucose is 6 carbons and ribose is 5 carbons. It'll just help you out with understanding what's going on later on. Okay. Man, when we talk, anybody in here had physiology already? Not enrolled in it now, but already took physiology. Raise your hand for me. Anybody? One person in here. So do you remember, you don't have to tell me anything that you remember about it, but do you remember learning about aerobic respiration? Yeah. Any of you guys ever learn about aerobic respiration? Metabolism. So you might have an idea, the two of you, of the importance of glucose in that role. Man, are you guys gonna love learning about metabolism. To me, metabolism is probably, and aerobic respiration specifically, one of the coolest things that you can ever learn about how your body functions. No joke. You're going to learn in my class, at least, and in physiology when you guys take it, because most of you guys will eventually have to take it, or you might be enrolled in it now, about why it is that if I were to smother you with a pillow, that you're going to die within minutes. And it's cool when you learn about how all that stuff functions and that it comes from your food, your energy. Of course, we know energy comes from your food, but the reason you need to breathe in oxygen is because you eat food to get energy. And when you exhale carbon dioxide, CO2, the O2 that's in the carbon dioxide is not the O2 that you breathed in. It didn't even touch that. Y'all gonna learn how that works. And I think it's some of the coolest thing. That CO2 that you're breathing out came from your food. Your food, y'all. So that is so cool, and I can't wait to explain it to you in chapter 10. Okay, but if you can't get the understanding of the fact that a carbohydrate has all these carbons and oxygens and hydrogens, that we have 6 of them, then when we get to that chapter, it's going to be a lot, because man, is that some chemistry, y'all. Okay, all right, so that's carbohydrates. Lipids. So lipids are, most of us know that lipids are, we can relate that to our fats, and that's great. That's a great place to start. So if you can relate it to your fats, then you can relate to the fact that it is hydrophobic. And so if it's hydrophobic, because we already know fat doesn't mix with water, it has to be hydrophobic. It looks like it's hydrophobic. It acts like it's hydrophobic. Then it probably has a whole lot of carbon hydrogen chains, and it does. And that's the most important aspect of these guys. We talk about carbohydrates and how their monomers are saccharides or sugars. There's no true monomer for our lipid categories, okay? So don't worry about it. The big take-home message that I want you to remember from our lipids is that they are hydrophobic. That's our big take-home message for those guys, okay? We talk about some groups of these guys, the triglycerides, that are basically going to be storage molecules for us. I don't need you to memorize. I'm not going to test you guys over what the structure of a triglyceride is, but do know that they fall in the lipid category, okay? So if you know they're in the lipid category, you know they are hydrophobic. Here's a picture of them. Isn't that nice? But I'm not going to test you over the picture, okay? You just know that they are lipids. They have hydrophobic. Same with the fatty acids. They got some hydrocarbon tails, okay? Here's probably the more important one that we will talk actually a lot about in the class, which is the phospholipid. Phospholipid is important. Is a phospholipid as a whole, is it overall hydrophobic? No, right, because what is it? Amphipathic, right, exactly, good job. So it has a hydrophilic head, which is charged, which is our phosphate group up here. Charged head, nice negative charge to it. And our long hydrocarbon tail, which is hydrophobic, no charge. This is really important because when you put this in water, those hydrocarbon tails don't want to have anything to do with the water. But does the charged head like being in the water? It sure does. So those tails are going to hide out away from the water and put those heads on the outside to interact with the water. So this structure that's over here with our phospholipids and the heads on the outside in the water and the tails on the inside. Do you guys remember what that structure is called? Well, almost. So one below it we'll call a phospholipid bilayer. This structure that it starts to form just from hiding the tails inside there, that round structure, it's called a micelle. You guys ever heard of micellar water? So the reason that it's so much better for your skin than just straight up water is because a lot of your skin has phospholipid bilayer, which exists with these phospholipids. So we have a lot of those in our membranes in our cells. So to make it a little bit more friendly to your membranes of your cells, we put in these guys that can interact a little bit better with your membranes rather than just freaking out your membranes with just water around it and all these charges and everything like that. So we'll introduce a little bit of that hydrophobicity in there with those micelles. That's the idea. That's like the basic version of what's going on with it, right? Because your phospholipid bilayer is a large structure that's made for the same reason. Put charged heads on the outside and hide the tails on the inside. And we made a whole 2-layer structure of that with tails on the inside and heads on the outside. So now the water base on the outside and the water base on the inside are happy, and we keep the tails on the inside of those 2 layers, make our membranes out of that, and now molecules don't want to go in or out because it's weird to have charges on the outside and those hydrophobic on the inside. Things don't want to pass through that. In fact, some of the only things that can pass through that are very, very small molecules that are charged or water. And I want you guys to think of it mostly just being water that we're passing in now. Because when we get into tonicity, man, that's going to help you a lot. Just thinking only water can pass through that, unless we put channels in there that are special made for things to pass through, right? So this is what keeps your insides inside your cell and your outsides outside your cell. Those are basics for that. We also have structures in our membrane and our lipid bilayer that are going to help give structure and shape to your cells. okay? We don't have cell walls that give us that shape like plants do and like bacteria have. We'll learn about this in chapter 4. But we have sterile base molecules. These sterile base molecules have all these rings in them. And again, I'm not gonna make you draw them or recognize them or anything like that. But here you can see that, of course, these rings are gonna give some rigidity to it. And of course, this hydrocarbon BS down here is gonna make it easy for it to interact with those lipid tails on our phospholipid, right? So that's how that's gonna stick in your membrane. But it's also why if you take cholesterol, which is our main structural component that gives our cells shape and structure. Why, if you take it and just put it in your blood, what's your blood mostly made of? Water, right? Do you think that water, which is polar, likes interacting with that BS? No. So that's why those guys will stick together and clump together in your blood vessels instead of mixing up with the water into solution. So they'll stick together and they'll clump up and get stuck in some of your more fragile, thin blood vessels and your capillaries and things like that, especially the nice thin arteries that are supplying the blood to your heart itself, your coronary arteries. And so it'll clog up in there and create blockages. And don't get me on my healthy either. So we have the sterols that have those similar structure, cholesterol being the one that provides our cells structure. We also have things like waxes. Obviously, wax doesn't mix well with water. We kind of know that already. It's kind of why we wax our cars, to prevent it from getting water beating up and everything that's not in, like rain that falls on your car. That stuff is not actually pure rain. It's not just pure water that's falling on your car, right? It's all the pollution and the dirt and the road dirt and everything like that. Let's keep everything that's mixed up in that water off of your car. So we give that hydrophobic coating on it to help it just feed off and fall off of your car. Waxes we know are hydrophobic. I'm not going to get into too much on specifics on waxes. We'll talk about a specific wax that I do need you to know when we get to chapter 4. All right, our proteins, these are the things that do a lot of the work in our cells. These are easily the predominant molecule that are doing a lot of the work in your cell, easily. So these are made up of our monomers for these guys. These do have monomers, so back to that, okay? These guys are made up of amino acids. And those are our building blocks. We have 22, I guess, that exist in nature, and there are 20 main ones that exist in human nature, okay? And these guys are linked together by something called a peptide bond. Let's see if I have a picture of it. Here's a list of all the 20. I will not ask you about them individually. You don't need to memorize them or their abbreviations or whether they're polar or non-polar. This is just giving you a sense that of all of those amino acids that we have, look how many different kinds there are, whether they are polar, whether they're non-polar, or whether they have a literal charge to them. Now you can start to see how those hydrogen bonds are going to come into play later on. Because if we have a whole slew of amino acids in a sequence that are non-polar, water-fearing hydrophobic molecules, they're going to want to hide on the inside of the molecule, whereas those charged guys might want to be on the outside. So we're trying to see how come those proteins might fold up the way that they do. All right, this is the basic structure of your amino acid. We have in every amino acid, we have the alpha carbon, which is the one that's in the middle, that is linked to an amino group, which if you don't remember what amino is, it's NH2. This is really good drawing. And that carbon, because remember, carbons always have 4 bonds, so we'll give it its 4 bonds to start off with. It's also linked to the acid part of the amino acid. So there's an amino and there's an acid. This acid happens to be carboxylic acid. If you don't remember from organic chemistry what carboxylic acid is, this is it. So this is the acid part of an amino acid. And this is the amino part. Okay. And then this is what makes it the individual amino acid, whether it's alanine, phenylalanine, tyrosine, tryptophan, whatever they are. This is the R group is what determines what it is. Okay. So there's some examples on the right side. So you can kind of see what I'm talking about with those R groups. They're blocked out in the colors there. And so it matters whether it's charged, uncharged, polar, non-polar, whatever. Okay. Those are linked together, each one where you have the NH2 group linking into the next carboxylic acid group. That bond between the NH2 group and the carboxylic acid group for the next guy, that's called a peptide bond. And a peptide is a term for short chain of them. If you have more than 20 of them, it's a polypeptide. All right. A polypeptide does not make a protein. You could have thousands of amino acids linked together. If it doesn't really come together and have its function, it's not really a protein yet. Okay. We'll talk about the structures of protein and what makes what next. Our amino acid sequence, basically how that protein is spelled out, whether it's alanine, tyrosine, tryptophan, whatever, that sequence, that amino acid sequence is the primary structure. So now we've created a ribbon out of every single amino acid that your gene's coded for from that gene. All right, we'll talk about how that works in chapter 6 and how that's spelled out by your genes later on. Then our secondary structure, well, if this guy's charge, and this guy is nonpolar, and then there's a charge, nonpolar, you can start to see how those guys might not want to be near each other, how they might dissociate with one another and start, moving on their little axes and creating movement and structure based on the guys that are immediately next to them. There's two main structures that you can get from those immediate interactions next to each other. That is the alpha helix, which is why I went to the next slide. It's the alpha helix and the beta pleated sheet. And essentially, these sheets are connected still by others that don't quite fall into these pleats, but they'll go back and forth and back and forth. And they're just kind of flat areas. You don't need to worry about the chemistry of why they do those things, but you do need to know that the alpha helix and the beta sheet fall in the secondary structure. The secondary structure is dictated by the R groups that are immediately next to each other. Okay. The tertiary structure. So now we've curled our ribbon. We took our ribbon, that was our amino acid sequence, the primary structure. We've curled it with our scissors. Now it's got curls in it and areas where it's not curled as much. That's our secondary structure. Now we're going to tie our bow. That's our tertiary structure. So now groups from across the way, these alpha helices and these ones over here, they're positively charged mostly over here and negatively charged over here, so they're attracted to each other. These guys are all nonpolar here, so they want to hide on the inside. They don't want to have anything to do with the water on the outside. All of the big picture folds, tying the bow, that's the tertiary structure for your polypeptide. Okay. So then what's the quaternary structure? What's left if you've already tied your bow? Well, if you're decorating your box, maybe you want to put more than one bow together in order to make it really look nice. In order for you to have your nice finished working functional product that you need, you're going to need more than one bow put together typically. Sometimes one bow is enough. But if you need more than one bow, that's your quaternary structure. Okay. All right. Enzymes are basically molecules that are going to help with chemical reactions, called them catalysts. They're overcoming the amount of work that's needed for a reaction to happen. So enzymes often do that, and they're not used up in those. We're going to talk about those in chapter 10, but that's a general idea of what an enzyme is. Antibodies are just proteins that are made to recognize foreign things that your body needs to label as foreign, so it can decide if it needs to get rid of it or not. That's basically what an antibody is. They're proteins. And based on how those proteins, the amino acid sequence is, how it curled the ribbon, and how the ribbon was tied, and how it associates with its other ribbons via its function, that's its native state on how it's made to be at its pH 7.35 to 7.45. That's the native state. Once we have too much acid, or we start adding heat and we start disrupting those hydrogen bonds from those attractions, we're denaturing that. We're removing this normal functional shape and therefore its actual literal function. That's denaturation. Talk about what role that plays in controlling microbes later on. All right, finally, we have our nucleic acids. You guys already know DNA and RNA. We have deoxyribonucleic acid and ribonucleic acid. So DNA constitutes your genes. DNA and RNA both consist of nucleotides. We kind of paint a picture of what that is. We have a sugar, which is our ribose or deoxyribose. We have a phosphate group, nice negative charge to it. Those alternate on the backbone, and then attached in the middle, we have our nitrogenous bases. I won't make you know the difference between purines or pyrimidines. We're not learning. I don't care about that in this class, okay? But you do need to know that they are called adenine, thymine, guanine, and cytosine, ATCG. Again, I'm not going to test you on those, but if we start talking about them later, be familiar with them, okay? And you do need to know eventually, for chapter 6 anyways, that A binds with T and C binds with G. The way that I remember which ones bind with which, the C looks like the G. And the T has harsh lines like the A does. And so there, the harsh guys go to the harsh guys and the round guys go to the round guys, okay? A binds with T, C binds with G, okay? And that's the gist of how your double helix exists. Again, I'm not gonna quiz you on that part. RNA is the same basic structure, but it just has true ribose instead of deoxyribose as its sugar. And it usually works as a messenger. We'll learn about how that functions in chapter 6. We're not gonna talk about necessarily here. They have major functions within your cell where your DNA is just storing your genetic information. Then we have ATP. And then that ATP molecule is our energy storing molecule. talk about this a lot in chapter 10, but you see how it has these 3 phosphate groups here? Man, look at a phosphate, you guys. This is a phosphate with these 4 oxygens around the phosphorus molecule. Look at those oxygens. Oxygen is always a Barbie hogger. Oxygen is always hogging electrons. Oxygen is always going to have that negative charge. So when you have just oxygens flying off of all these different phosphorus groups, hogging those phosphoruses, electrons, we have negative to negative to negative, forced to be sharing electrons with each other because of those covalent bonds are there. And they're literally chained together and they hate each other. They don't want to be anywhere near each other. So they're all the time trying to get away, like south poles of magnets, pushing each other apart. So when you cleave off one of these guys, where you have ATP triphosphate, and you make it into ADP diphosphate, and you've cleaved that bond, boom, energy is released. This powers almost every single aspect of everything that goes on in your cell. In fact, the reason you die when I suffocate you with that pillow within minutes is because you run out of this. Everything that you're eating, of course, you're building things too out of the products of that, but the energy that you're getting from the food that you're eating makes ATP. The reason you suffocate and die is because you run out of oxygen that is needed for the process of making ATP. You need the ATP. That's what you're dying from. Okay? Because you run out of ATP. It's that important within minutes. That is impressive how important this is to maintain survival of a multicellular creature. So do not let that get lost on you as we go through these chapters, how important ATP is. It's not just some molecule you learned about in chemistry. The key to the ATP, and we talk about the key with the hydrogen bonds. Huge, huge things in biology. So we're already over time, so I'll go over the questions. Lucky you. Covalent bond, sharing electrons. pH is a measure of the hydrogen ion concentration. Be sure you understand how that scale works, right? Major and essential biological role of phospholipids. They make up our lipid bilayers for our membranes. The monomer subunit for proteins, amino acids. All right, that was the last one. Yeah. There we go. Okay, thank you. What types of molecules tend to be hydrophobic? Non-polar molecules. That's what I'm looking for that, okay? Or you could think, if you thought lipids, that's fine too, because they are non-polar, right? Okay, makes sense? Good. Any questions before we go to the lab? We'll start lab typically at 11:30. I'll be a little lenient on that if you need to go have a bathroom break or a smoke break or whatever. I'm going to pop up the Kahoot for the lab. You guys come in there and sign in when you get in there. If you want to just go straight up there, I'm going up there too. So I'll see you guys when you get up there. It's a pre-lab. So you're supposed to so they are not review of the previous lab, but they're questions about what we're going to be learning about in the lab. 99% chance I'm going to make them participation based. Right now, how I originally had it set up, and I think I have it in the syllabus set up as being 4 credit, but it's probably going to be participation based. I just don't feel like putting that pressure on you guys. But as long as you guys are putting forth the effort to prepare for the lab, that's the idea. So I'll probably be pretty lenient on it, but if I start seeing that people aren't really prepping for the lab, I reserve the right to, yeah. Yes, what's up?
I sent you an email. I didn't